Hi, everyone. Thank you for joining the Family Office Network's webinar series. My name is Lindsay Shuck. I work with Family Office Networks. Today, I'd like to introduce our speaker, Dr. Mark Allen, co-founder and CEO of Elevian. Mark is a medical doctor and successful serial entrepreneur. He has partnered with 3 Harvard professors and a team of drug development veterans to found Elevian. Mark, over to you.
Thank you, Lindsay, and thank you everybody for joining today. Before I get started, just wanted to say that there are an ability to ask questions. If you have any questions as I'm going along, please type them into the box where it says questions, and I will get to all of those at the end. There should be plenty of time to review any questions. So wanted to start with really 3 points that I wanted to get across. Number one is that aging is malleable. The rate at which our bodies biologically age is subject to intervention in both directions. We can do things that increase the rate at which we age, and we can do things that slow or even reverse the rate at which we biologically age. Number two is that a little known protein called GDF11 plays an important role in the aging process. And number three, that Elevian, who is working on developing drugs that impact the GDF11 pathway, and as we really discovered the role of this pathway in the aging process and have quite a moat of IP around this, is an exciting investment with the opportunity to bring new drugs that can change the face of humanity. So what is 50? You know, I just turned 50 a few months ago, and I've never felt better in my life, energy-wise, physically, mentally. But you know, it was crazy. When I was a teenager, I thought that this was 50. If you guys recognize this, Wilford Brimley, when I was a teenager, starred in the movie Cocoon. And this picture is Wilford Brimley at the age of 50. I thought this was ancient. You know, with superpowers coming from an alien race, the greatest stunt that Wilford performed in that movie was to jump off of a diving board. Well, this is 50 today. Tom Cruise, so this was actually a few years ago, but he's still doing his own stunts. So, you know, this just shows what we can become. Now let's look at 80. So 80 today is the average age approximately of death, and this is what many 80-year-olds look like: multiple different chronic disease, functionally debilitated. But with simple interventions available to all of us, this is what 80 can be. And this is Ernestine. So in her 40s, she looked at the mirror and saw somebody flabby looking back at her, began exercising, and ended up winning bodybuilding contests in her 60s and 70s. She's now, I think it's 82, and running a fitness center. This is a picture of her at age 80. So this just points to the fact that aging is malleable. Now, one way that we know that aging can be changed, can be intervened, is in young blood tests and blood exchange. So our co-founders worked in this model called parabiosis, where they conjoined the circulatory systems of young and old mice, and leaving the mice together with blood flowing freely between these organisms for 4 weeks straight. They discovered that old mice exposed to young blood experience a biological reversal of aging. By many different measures, they see the organs growing younger. And interestingly, the young mice exposed to old blood have the opposite effect. They have accelerated aging. So this implicates circulating factors sending messages back and forth between these two young and old organisms. So our group was really the first to ask the question, what might these factors be? How do proteins change with age? And what proteins might be sending these regenerative and degenerative signals? So out of that analysis, they discovered this little known protein called GDF11 that by many measures decreases with age. They got access to a recombinantly produced version of that protein, and they estimated what it would take to replenish GDF11 levels in the aged animals to youthful levels. And much to everybody's surprise, by just injecting this single protein, they were able to reproduce many of the regenerative effects of young blood. So in studies that were rapidly published, they showed that GDF11 can rejuvenate the aged brain, can restore patterns of youthful vascularization, and stimulate new neuron production. In the aged heart, can remodel the heart to a youthful state. And an aged skeletal muscle can restore the regenerative potential, how rapidly skeletal muscle heals to a youthful state. So these studies were published in Cell and Science, not bad journals, and were recognized as a top scientific breakthrough. So this is the group of our co-founders that worked on these studies. So 3 professors and a postdoc working at the Harvard Stem Cell Institute. And I partnered with these folks and put together a team of drug development veterans who have collectively brought dozens of drugs to the clinic and produced over 4 billion in exit value. So Tony Sandrasagra, who leads our R&D efforts, was formerly chief science officer of another spinout from Rich Lee's lab, has about 25 years of drug development, both in big pharma and in smaller biotech. Manisha Sinha was the first author on that muscle regeneration paper with GDF11 while she was a postdoc at Amy Wager's lab, and spent about 5 years in industry before we recruited her into Elevian. Orit Ford was formerly the head of antibody therapeutics at Stemcentrx, which was a huge financial hit. And Yangting Wang was one of the leaders of one of the leading labs studying stroke preclinically. So Elevian today, we've been around for about 2.5 years. We've raised about 9 million to date. Today we have 8 full-time employees. We're located at a facility at the Harvard Life Lab. So it's part of the Harvard Accelerator program affiliated with them. Have about 4 contractors and many different collaborators and a number of world-class advisors. So we have collaborations at Harvard and Duke today, and Silico Medicine, and several CROs. And we're working right now on raising a round that will help us to initiate our early clinical studies in humans. So one of the unique things about Elevian is that our founders were so early to understanding this pathway. So most drug companies are building a drug that targets a known pathway. So this is really the first drugs and first approaches in this GDF11 pathway. So little was known about this protein because it circulates in concentrations that until recently were not detectable. So as a result of this early work, our co-founders in Harvard University were able to patent quite broadly patent claims to use the natural protein, GDF11, to treat and prevent age-related diseases, as well as a number of new compositions to upwardly modulate GDF11 to treat a variety of different diseases. So following the initial studies from our co-founders, now our lab and more than a dozen other labs have shown that the same doses of GDF11 can be used to treat animal models of all of these different diseases. So this is quite amazing to have therapeutic benefits across the body like this. GDF11 appears to broadly stimulate a healing response that can improve disease and prevent disease potentially across a number of different areas. So all of this is animal data. So I say potentially because we need to translate this into humans. So out of all of these different diseases, the heart, the brain, the lungs, cancer, kidney failure, liver failure, diabetes, our big question is, what is the right approach to market? We can't tackle all of these at once. So what we've done is very carefully selected a couple of areas where we see effects very rapidly against devastating disease. So where there is a clear commercial need, where there is a regulatory path, and where there's compelling data to support. So we've selected 2 different areas. So number 1 is the area of stroke recovery, and number 2 is the area of metabolism, diabetes, and obesity. So I'll go into these in a bit more detail. So first of all, stroke. So stroke is worldwide the #2 cause of death. About 650,000 strokes that are clots per year in the United States. And the only drugs available to treat stroke, there's one drug and one device that's been approved that eliminate clots, but they must be given in the first few hours after a stroke. So they're approved to about 3 hours, and standard of practice, they'll use them up to 4 hours after a stroke. So as a result of the time window, and as a result of contraindications such as high blood pressure or history of bleeds or an anticoagulant, most patients do not get this therapy, and many that do still have ongoing deficits. As a result, stroke is the #1 cause of long-term disability. We all know people who have had strokes that dramatically affect their lives. So what really compelling here is that GDF11 appears to work very well as a way to improve recovery after a stroke. So the initial data and understanding of this came from our co-founder, Lee Rubin, who discovered in these aged mice models that GDF11 can improve the vascularization in the brain of aged animals. And then we think secondarily to that, it stimulates new neuron growth in neurogenic regions of the brain. We say secondarily because Lee has done some work understanding that GDF11 does not appear to cross the blood-brain barrier. So its primary activity is improving that vasculature, which of course you improve the plumbing, you bring good factors in, bring the waste out, that can cause a lot of good. So since our initial studies, now 3 different independent labs have published studies showing that GDF11 can promote recovery after an ischemic stroke. So the injury model here is a transient occlusion of what's called the middle cerebral artery. That's the most common location for a stroke in humans. And these 3 different studies, one was in a young mouse, one was in young rats, and one was in old mice. And they used what we consider from many different studies to be the low end of a therapeutic dosage range of GDF11, injected once daily for between 5 to 7 days. And in all 3 of these studies, they were able to replicate the improvements in vascularization and neurogenesis, and very importantly, show that GDF11 given after a stroke can significantly improve motor function recovery. So very exciting finding where, again, there is no treatment to improve motor function recovery. So really interesting is the young mice, most of these mice survive the stroke injury, but in the old mice, many of them die from the stroke injury. So in those models, we see also an improvement in survival. So let's take a look at this data a little bit more. So in the improved survival data here, so this is a survival curve, and this dotted line shows without treatment. So these animals are injected with the vehicle, but not the drug. So it's a control animal. And in that, about 40% of those animals die. But when those animals are given GDF11, most of them survive. Only 10% of the animals die. So, this is really significant, but we don't want to just save people, have people living that are non-functional. So it's really important that we also improve that motor function in animals. So this is showing an assay of motor function recovery. This top line here is a sham surgery. So the animals are opened up, but they're not actually given a stroke. This bottom line is vehicle-treated animals. So animals that have had strokes, but not the drug treatment. This middle line is treatment with GDF11. In this particular study, the treatment was started 7 days after the stroke and went on for 7 days. So at 7 days after treatment, you start to see a separation, which becomes significant at 28 days and continues ongoing. And you see as these animals get out past a month, the treated animals are not significantly different than the normal animals. So this data coming from independent labs and built on top of our IP claims, it was really exciting to us and made us think, wow, we could give just a few days of treatment potentially of GDF11 and significantly improve clinical function in humans. And so we began to think, let's take this really pilot data from industry, and let's take a drug development approach to this and put it through the most rigorous possible models in stroke recovery. So we partnered with, we brought on board a leading group of advisors, partnered with the leading CRO in the world to look at stroke recovery models, and we used a model in rats of permanent ischemia. And so this is really hot off the press, where we see in a rat ischemia model that GDF11 can improve recovery across multiple different measures in as little as 4 days of treatment. So in this case, we started treatment 3 days after the stroke event. So that starts at normal function at day 0, comes down in both the treatment group and the vehicle group day 1 after the stroke, and then by day 3 there is some recovery that's natural in the animals. Day 3 we start treatment, and then you see a very significant separation between the vehicle and the treatment that continues out over time. So really exciting data. This is using a 10-time higher dose than those initial studies. We're now working on trying to figure out what is the optimal dose, what's the optimal treatment duration and treatment initiation to help to support our clinical studies in humans. So while we have not yet begun our clinical studies in humans, there is data from an independent lab out of UCSF measuring GDF11 levels in humans. Now, this particular assay cannot differentiate between GDF11 and a related protein called GDF8. So this is more accurately the combination of these two proteins. But what this data shows, this is data from 1,000 patients in San Francisco and has been replicated with 1,000 patients in Norway as well. What this data shows is that humans that have the lowest quartile levels of GDF11 and GDF8 have a significantly higher risk of disease and death relative to patients that have naturally higher levels of these proteins. So this is quite significant. This almost looks like a dose response kind of data, but this is data measuring patients at the beginning of a 6 to 9 year study period and following them over time, and showing that GDF11 higher levels are protective against cardiovascular disease, stroke, and death. So this area of stroke recovery is a massive market. We would be one of the most valuable companies in the world with a successful treatment for stroke. But I want to point out that this is really only the first application of many for recombinant GDF11. So I'll talk to you about some of the additional applications, and then we're also working on other drugs, other ways of modulating GDF11, either drugs that could be taken orally or injected far less frequently. So one of the very exciting ways that I mentioned is this area of diabetes and obesity, so the general area of metabolism. So now there is data suggesting that GDF11 mimics calorie restriction. Calorie restriction is one of the most studied ways to intervene in the aging process using many different models. Calorie restriction is shown to reduce morbidity, reduce the risk for disease, and improve longevity. And what GDF11 appears to do is to provide a healthy, durable weight loss across many different models and improving overall glucose metabolism. So this has now been replicated in many different labs, including our lab and the Harvard lab. So really exciting applications here as well. So we have a development plan that really brings us out to our clinical trials. So talk to you about really where we are right now. So our first drug that we're bringing to market is recombinant GDF11. So we've figured out a proprietary way to manufacture this drug to express the protein using mammalian cell lines and in a scalable process. So today we are producing, to our knowledge, the highest quantities, the highest yields of GDF11 of anyone in the world. We are taking our process and we are right now working to select a contract manufacturer to scale this process and put it into an audited GMP process to produce drug that we can use for comprehensive safety and toxicology testing first in animals and then in humans. And that's really the process to get to human testing, is we have to take that audited process to produce a drug that we use in what are called IND enabling studies and our human phase 1 safety test. So that same process can be used to support multiple different applications of the drug. So while we're doing that, we are continuing to study really what are those best applications. So I talked to you about stroke recovery at a very high level, diabetes, obesity. We're also looking at some orphan indications that may provide an even faster path to market. So ideally, we will have, with sufficient data and enough money, the opportunity to do multiple phase 2 studies in parallel, based upon the same phase 1 study and IND enabling studies. So last, we have a number of discovery efforts underway to find novel molecules. So the antibody program we're making quite a bit of progress there, and antibodies can be injected less frequently. We're also working with one of the leading AI small molecule discovery vendors to identify small molecules that can be formulated into pills, and have other ways to work on GDF11 formulations to create slow release that we're working on as well. So a number of different ways to ultimately get to even better drugs. So I wanted to share some perspective of competition. So we are not aware of any other groups that are working on GDF11 as a therapeutic. We have quite a moat of IP there, but there are a number of companies working on drugs that broadly target aging with that same hypothesis that one drug or slight variants of the drug can be used to treat and prevent different diseases. So perhaps the leader in this, certainly the leader in terms of valuation, is a company called Samumed, which is biologically similar to GDF11 in that they're going after a single pathway called the Wnt pathway, whereas we're going after GDF11. So Samumed is quite far along in that they have a couple of programs in late phase 2, beginning their pivotal trials, one for male hair loss and another for osteoarthritis. And they're the world's highest valued private biotech company. And really that valuation is a result of the addressable market that's available when you go after this broad aging space. Denali Therapeutics is going after neurodegenerative disease, have a couple of phase 1 programs, a public company, quite highly valued. And Unity Biotech is a company that is selectively targeting zombie cells, so cells that stop dividing but don't die and release inflammatory factors. So they have a drug that is systemically toxic that can destroy these senescent cells, as they're called. Because it's systemically toxic, they need to inject it locally, and their first program is injecting it into the knee to treat osteoarthritis. So this kind of shows the valuations of these kinds of companies. Now, the investment in Elevian, you're really getting in on the ground floor of the company here. We have our current round is a 40 million pre-money valuation. We're raising 7.5 million. We've got 4.3 million in the bank right now. We have Lauder Partners, Save Earth Fund, some institutional investors, Tony Robbins coming into this round, and also partnering with the company. He's very excited about us and putting us in front of many of his clients. The caveat here is like many private investments, this is for accredited investors only. So with that, I wanted to open it up to questions, and thank you very much for your time. Okay, so I'm just reading out these questions. Let me see. So the first question is, can I share the approximate population size of the animal tests? Also comment on the FDA requirements and status with them. So the animal tests that I shared were of different sizes. So I think I showed in the survival test that was an N of 21 in each of the groups. Ours was a little bit smaller in the rat study. Yeah, some of the published studies were even smaller than that. So those kinds of numbers per group, 10 to 20 kinds of numbers per group in those animal studies. We're doing a much larger study now, looking at toxicology, using more animals than that. And a comment on the FDA requirements. FDA, so the FDA requires that we do, in order for us to begin human clinical testing, okay, that number one, in the area of stroke, you only need efficacy tests in rodents. In many other indications, you need efficacy tests both in rodents and in some second species. So stroke is kind of unique in that there's not great models in larger species, and because it's such a devastating disease with so little available to treat it, we're allowed to get into clinical trials with only those efficacy studies. Now, regardless of indication, we must do safety testing both in rodents as well as in larger species. So that's what's necessary, and we must use a drug that's in an audited process in order to do those. Okay, so let's see, the next question. Okay, where we're sourcing the protein for production. Okay, so the protein that we are making today. So we have developed proprietary cell lines. So we licensed a mammalian cell line called Chinese hamster ovary, or CHO cell. It's an immortal cell line, comes from a Chinese hamster's ovary. Quite interesting. So we have used a virus to insert the GDF11 gene into those cell lines, and we use a proprietary sequence of the gene to optimize expression and post-translational modification. So this is quite a complex protein to produce. So it requires sophisticated refolding and then enzymatic processing afterwards. So we developed that cell line, and we're taking that cell line into a contract manufacturer to effectively scale the sizes of the batches, the conditions to maximize yield. And then we also have developed a proprietary purification process as well. So the next question. So the cost to get through the various stages. So the cost for us to get into from where we are now to get into human clinical trials, we estimate to be about $20 million. Just call it $15 to $20 million to get into human clinical trials. But to get through human clinical trials can easily be upwards of $100 million beyond that. Okay, so the key is that being a biotech company, so most of companies in our space will go public well before they enter the market. So they'll either tap the public markets, or they'll do partnerships with pharma companies to help to support the later stage clinical trials. And so I showed you some examples of a company that went public in preclinical stage, a company that went public in phase 1, and a company that is in late phase 2, beginning phase 3, that's not even a public company. So there's really a lot of optionality there. What we're interested in doing is, as a company, providing a path to liquidity for our early investors that could either come from the public markets or could come from later stage investors that are interested in keeping us public, interested in buying as much as they can and being able to offer that liquidity to early investors. So that path to liquidity, while it could very well take us a minimum of 6 years, let's be clear, to get to market, we could have a path to liquidity in a 3 to 5 year time frame. So the next question is, ischemic stroke is a very challenging indication to go after, with many companies having failed. So why wouldn't we start with diabetes, obesity? So let's talk about ischemic stroke and where those failures are. It's an excellent and perceptive question. So the failures in ischemic stroke have been in targeting the acute stage of ischemic stroke. So where there is already treatment, and extending the time frame for either that treatment or providing other ways to improve functionality within that in that very acute stage. So there's been quite a number of failures there. And what we are doing is quite different. So we're not treating in the acute phase. We are waiting, we think clinically, at least one day, waiting for that patient to be stabilized where it's no longer a medical emergency, and where there's not existing treatment. And we would then begin treatment in that time frame. So there is no treatment there. No drugs, very few drugs that have been tried in that area, and that's where we're seeing an effect. So we think that we're doing things quite differently than what has failed in ischemic stroke, but we'll note that there are absolute challenges in the area of stroke. So there's quite a big variability in where humans have strokes. What the deficit is from what appears to be the same stroke injury in imaging could cause different deficits and different rates of recovery. In general, the healthier somebody is, the younger somebody is, the more potential they have for a more significant recovery. But all of that variability makes it harder to show an effect with a drug. So we are very carefully looking at how can we segment that population to reduce the variability, find more homogeneous types of strokes in imaging, in clinical deficit. And so we've done quite a bit of thinking about those populations, and even to the extent of specific subsegments of stroke that are considered orphan disease. So happy to share more details of that under NDA. And the other thing is diabetes and obesity. So diabetes and obesity, the challenge there, we do think that there is some compelling data. We are a little bit behind in terms of building an advisory board there. We have an excellent stroke advisory board, just starting to build our diabetes, obesity advisory board and really understand what patient segments to go after. The problem with diabetes and obesity is there are many treatments already available. So we know that while there's a great endpoint, weight loss and hemoglobin A1C there, the trials that we need to do need to be considered against gold standard. And so those trials are naturally going to be a lot bigger, a lot longer, looking at safety over time because there are existing treatments. So it will be a lot more expensive clinical trials to work in diabetes. So next question is how do you scale that? If you can answer a little bit more, I'm not sure what you mean by that. So how do we test for human GDF11 levels is the next question. So there are a number of ways to treat, and we are working on developing a cost-effective companion diagnostic. So today they're all research grade. So the study that I mentioned is using an aptamer. So it's a DNA-based test that has very strong, very high potency binding to GDF11, but the problem is that it doesn't differentiate between GDF11 and the closely related protein GDF8. So we as a company have been developing a number of different, and right now there's no perfect way to detect GDF11 selectively, and the other problem is it's confounded by both close. GDF11 is very closely homologous in structure and in binding sites to GDF8, and shares some binding sites with other serum proteins as well. So finding something that's selective is really a key thing. And the other problem is that GDF11 exists in a variety of different molecular states. So we've done quite a bit of work over several years and inherited some work that was done by our Harvard co-founders to identify selectively GDF11. So we're making a lot of progress on that. And at the time that we have ourselves start our human clinical trials, we expect to have a cost-effective companion diagnostic to very accurately measure GDF11 levels. So how confident are we that there's not another company with a similar product in later stages of testing or posing risk due to a comparable but more advanced solution? So, the answer to that is it, there could be somebody that just hasn't published that's doing some interesting work in this space. That is always possible. There's nobody that's been published. We're on top of the literature, and in the literature, they always mention are their industry interests in there. You must disclose that. So we're not aware of anything coming out of the industry, coming out of any information that we have from scientific conferences. The I would say that if there is somebody working on this, then they are likely to bump into our IP. So we were very early. We are very well aware of what's being published in terms of patents. We're continually staying on top of that. So we're not seeing anything that we're concerned with, and vice versa. Anybody who enters this space should be quite concerned with us. And then I'd say the last thing is, this is not easy science. This is really difficult science. We have, some of the best scientists in the world working on this problem, and so there's quite a bit of skills that we as a team have in our focus on this specific area. So next question, is osteoporosis a possible treatment here? And could this apply to cancer? So the area of osteoporosis, there's mixed data in its GDF11's effect on bone. So there's data that's been published in both directions. It's got a positive effect and a negative effect on bone. So the jury's out there. We'd have to study it a lot more, and so it's just not an area of focus, and relative to the other things that we're doing. So how could this apply to cancer? So let me touch on that. We had concerns, this GDF11 being a growth factor. So GDF11 and its family of proteins are growth factors. So GDF stands for growth differentiation factor. So with any growth factor, one must be concerned about whether it will cause cancer. Now, we know that GDF11 broadly stimulates the growth of vasculature. So, that was quite a concern. And more than 10 years ago, before our founders published their data, there was a study showing that GDF11 levels were increased in colon cancer, so associative. Now, since then, several studies have looked at interventionally, how does GDF11 impact cancer? And in several different studies, it has been shown that GDF11 has a tumor suppressive effect across several different virulent forms of cancer. Now, that tumor suppressive effect is not a tumoricidal effect. So there are drugs that target to kill cancer, or help our immune system kill cancer. This doesn't appear to be that, and more of a suppressive effect. So we don't think that this would be a primary treatment. There's lots and lots of drugs in cancer, so we don't think that's the area to focus for this drug. Although it would be wonderful if it indeed had a tumor suppressive effect. Great positive off-target effect. So the next one is, what is the mechanism of action of GDF11? So, great question. And the mechanism appears to be different in different tissues. One of the interesting things about GDF11 that also I think a traditional drug development company, pharma company, would shy away from GDF11 development is because it has such broad activity. So many cells in our body make GDF11. There are receptors for GDF11 on many different cell types. Now, therapeutically, it appears that the primary mechanism of action is binding to the endothelial cells and stimulating endothelial progenitor cells to grow new vasculature. And we see the vasculature, both quantity as well as quality, improving throughout the body. And we're with our lead program in stroke, most interested in the brain and have studied that most significantly. But there are other studies showing the improvement in vasculature in the heart, improvement in vasculature in the periphery. Can show effects. There have been reported effects improving recovery after a heart attack, improving atherosclerosis, improving wound healing in a diabetic model. So, that we see that to be the primary effects. So the next question is, what is the target timeline, assuming that we're successful at raising the required capital to a possible exit? Okay, so the possible exits we see in a, call it a 3 to 5 year time horizon. Now, I would say that that would be the first point of exit, but if we are successful, the value of the stock will continue to rise significantly from there. So we as a company are thinking about not going public in a preclinical stage, but rather going public once we show a successful human proof of concept. So I showed that development timeline. So we first need to do a human safety study. Then we would do a phase 2 study, and an early phase 2 study, we would have early readouts where we would look for multiple different endpoints. And for example, we might do a study in stroke and a study in diabetes metabolism simultaneously, so that we could have effectively multiple shots on goal showing positive effects in humans. With that data, we could go public by many measures at well over $1 billion valuation. So that would be really the earliest time point that we see. Again, we could go public earlier, but I think that there's risk and distraction associated with that is what we feel. So the other thing is that there may be an alternative that would still provide a path to liquidity for our early investors, later stage investors coming in and wanting to buy the stock and keep us private would be also an attractive alternative to us. So a question is, how do we scale the protein production? Okay, so there are really a couple of factors there. So number one is the cell line itself. So the cell line, how much protein is being produced by the cell line? How dense of growth conditions can we grow that cell line? And then what size vats can we grow those cells in? How much media do we need to grow the same cell? So that's kind of one. It's just the expression of the protein, and then the second piece of it is the purification of the protein. So the process to take that media and pull just GDF11 out of that media. So we've been working on process design ourselves. In really research grade process design, getting it to as good as we can. We've just brought on board a leading protein biologics manufacturing expert to help us with this. And ultimately, right now, we need to go to the next step, which is to move to a contract manufacturer. If we were to build our own manufacturing facility, it'd be about a $10 million capital investment. So most companies in our stage instead work with a contract manufacturer who produces drugs for multiple different companies. And there are several that focus only on protein biologics. So we are interviewing several of those companies and going to select really one partner to work with initially. And they basically put it into much larger vats. They use a bioreactor where they can carefully measure pressure, temperature. They have lots of different techniques that they use to scale it. So next question is great to see all of these great questions. The next question is, how do we plan to use an antibody to GDF11? So interestingly, our antibody is not to GDF11. Our antibody is to a natural antagonist of GDF11. So GDF11, while I mentioned it circulates in extremely low concentrations, it is also highly regulated. So there are a number of natural antagonists to GDF11 and closely related proteins. So, it just indicates just how important this protein is in the human body. And by the way, the GDF11 protein is 100% identical across mammals in its mature domain. So our antibody is to a specific natural antagonist of GDF11 that interestingly goes up with age. And when we measure this antagonist in humans, we find that it has the opposite effect as GDF11 in humans, in that high levels of this antagonist predict increased risk of disease and death in humans. So we have already discovered an antibody that neutralizes the binding of GDF11 and this antagonist. In other words, it can break the bond of GDF11 to this antagonist, free up GDF11 to activate signaling. So an exciting candidate, but we have not yet begun our in vivo, our animal model testing with that candidate. Next one, is there a relationship between blood types and Rh factor with GDF11? Not to our knowledge, not to my knowledge at least. So interesting question. So what has stopped us from receiving additional funding from different VCs in the area so far, and what have the objections or concerns been? So what's interesting is that we are doing very different type of drug development than what is natural. So, a typical asset-based company, there's kind of two companies in the area of drug development. There's asset-based companies and there's platform companies, and we're kind of between the two. So, a platform company generates lots of different assets, some new therapeutic modality, mRNA-based therapeutics is one that came out several years ago. That can generate lots of different molecules using their platform and license them out early, so have early revenue, don't have to pay for the drug development costs. So that's one type of company. And then the other type of company is an asset company. Usually an asset company has maybe one or a couple of different assets, and the asset companies typically have a very high risk of failure. So the drug either works or it doesn't. And so the VCs so let me say, what we are as a company is we're a mixture of the two. So we're a big biology play around the GDF11 pathway. We can, in this pathway, we're producing multiple different assets and have the IP to support that. And any given asset can be used to target multiple different diseases. So effectively, we are a portfolio player ourselves. Now, of course, it will require a lot of money to target multiple different diseases. So, most venture capitalists, biotech venture capitalists that invest in assets say to us, pick one disease. Let's just focus on one disease. Well, the problem with that is they're the portfolio player. It's okay with them if we fail and go out of business because they've got 9 or 19 other investments that could potentially succeed. We don't want to fail, so we want to build the company to be able to go after multiple assets. So we are bringing in, we have several institutional investors. I kind of skipped through that quite quickly, that are already in the company. We're bringing more into the company, but it's a little bit different of a profile, and something that is more attractive to the limited partners who are interested in the diversification than I'd say a lot of the venture capitalists. So is this angiogenic? So yes, yeah, as I mentioned, very much so. It stimulates both the quality and quantity of vasculature, although it appears to do that in a healthy and beneficial way. And I think that the secondary question, does it feed blood supply to tumors? So again, that was our concern, but we see in tumors, we see a tumor suppressive effect. So, well, if it is improving blood supply to them, it's also improving the body's ability to fight. Question is, what was the pricing of the previous round? What type of securities issued? Are there any VCs committed to this round? So the pricing of the previous round was a safe note at a 44 million pre-money valuation or a 20% discount, and that is converting into this round. So this is a preferred equity round. And the VCs committed to this round. So family office / VC Lauder Partners is in this round. There is an institutional investor VC called the Save Earth Fund that is in this round. Those investors are already committed, more than committed. Have already provided funding into this round. So is there a more detailed prospectus? There is not a more detailed prospectus. Certainly there is a lot more data available that we have for you. A lot of that data is available under NDA. We do have a data room set up. And so, happy to share more data. So several of our investors have accessed the data room. So if you'd like to talk to those that have done more extensive due diligence, I'm happy to make some connections there as well. So that looks like the end of the questions. Thank you all very much for all of your questions and your interest. And if you have more interest, you can reach out to me directly. I've got my email right there, mark@elevian.com, and look forward to hearing from you. And thank you again for your time. Bye-bye.